So as you have nicely set the stage about why we need to know about biosimilars and how we need to see how we choose them. So I'm going to go through the topic, and hopefully in the end we will be a little wiser as to how we choose this biosimilar. Now, we all know what a biosimilar is, and the thing we need to remember is that uh, there are definitions given by FDA and EMA as to what biosimilars are. We also need to know what a biosimilar is not. So we have a term called biobetter. We have biomimics or biocopies, uh, and of course the generic drugs. So biosimilars are definitely different from biobetters or biomimics or biocopies. Now,
Okay, sir.
why biosimilar is
Thank you. Thank you,
because,
sir.
as this slide shows you, that the main purpose of having biosimilars is to decrease the cost and increase the uh, reach for all these biologic drugs to a larger percentage of population. Because as the slide shows you, that the assumed year-on-year growth for the approved biologic is about 10%, and the biologic penetration is about 60%, and the biologic price discount, what is being told is should be about 35%. So these are the advantages, and that is why we need to look at biosimilars. Now, why a biosimilar? The challenges is that it is usually reserved for later line of treatment because of various reasons. And with the cost savings that we have, we can look at using them in earlier lines of intervention. As I mentioned, we can increase the access to cost. Additional budget is then available for innovative therapies. And because of these biosimilars, since you're using it in rarer disease and niche indications, you're actually producing more data and also having additional budget available that can be allotted for better research. Again, this is just to show you that biosimilars in have a moderate sort of costing there. Biologics in the US are very high. Generics are low cost, and the development cost is what is the most important thing that goes into the costing of any of these drugs. And this is actually showing you the time frame that takes for a novel drug to be developed, which is almost 14 plus years, but for a biosimilar it is 6 to 8 years, as in contrast to a generic drug, which is much, much shorter, as to 3 to 4 years. Now, this is actually from a survey looking at how familiar are various stakeholders in terms of biosimilars. Overall, if you see, the red one is for not familiar at all. So if you actually see physicians, pharmacists, and nurses, which I believe are major stakeholders, the lack of knowledge of biosimilars is pretty high among physicians, which I was also surprised. But it seems much better with pharmacists. Of course, with nurses also it is pretty high. But I think what we need to remember is between physicians and pharmacists, there is a huge difference. So that is an important factor that we need to remember, and I will touch upon this again towards the end. So time for vigilance, choosing the right biosimilar is to ensure better patient outcomes. So few things we need to know about biologics is that they are complex molecules. They have sugars, proteins, nucleic acids, and complex combinations. They may be living entities such as cells and tissues that are involved, their natural sources, and biotechnology is the method by which all these are produced. So this is just to diagrammatically show you the difference between a biologic and a chemical medicine. This is for one of the simpler biologic entities, that is erythropoietin, and this is for aspirin, which is one of the simplest generic drugs. And even between these two simple molecules, you can see the huge difference in terms of the structure and the size itself. So that is an important thing that we need to remember. So going by the size, if you look at ibuprofen and you're looking at the number of carbon molecules there, it is 13. Whereas if you go to trastuzumab, it is 6,000 plus. So that is how complex these molecules are. Then if you look at the evolution, 1950s is when DNA was discovered as a molecule. From then on, we have the genetic coding that has been unraveled completely. We have the basic biologic techniques that came in 1970s, but from there has been a rapid development of all these techniques and outburst of all these molecules, the biologics that I've shown you here. And today, I think every year there are several biologic molecules that are coming out. So it's important that process is the product when biologics is concerned, and each step is crucial in ensuring quality of these biologics. So I'll touch upon a few of the steps that are important here. So this is the development approach for a novel drug. Uh, for a biologic, they look at the physicochemical characteristics, they have a biological characterization, there is some preclinical data, PK/PD, and then the clinical data. So this is what happens for a very new drug that is being developed, and this is from the Clinical Journal of Nephrology way back in 2014. So very important events are these two steps. So if you look at characterization of a biosimilar drug, the primary focus is on this structure because they need to have a foundation for establishing comparability. And this is how the structures are compared. This is the biosimilar versus the innovator. And as you can see, the two are mirror copies, and that's what must be reached for a biosimilar to be working as good as an innovator. The other thing is characterization of the study matrix for biosimilars, the physicochemical properties, and the bioactivity assays. And this is just a listing of the various steps that they need to look into. Now, monoclonal antibodies, which are essentially the biologics, can be characterized by using state-of-the-art analytical methods. And on the left side is all the biological characteristics, looking at each part of this monoclonal antibody. And on the right side is the physicochemical properties that are all enumerated. They need to look at the amino acid sequence, the oligosaccharide, there's a disulfide bonding, and the C-terminal heterogeneity as well. So they're looking at the fingerprinting. This is the reference biologic. The biosimilar must have a very, very similar fingerprinting as shown here. So this is the difference between the development of a biosimilar and an innovator brand. So here, for the originator brand, you see that they need to look at a large proportion of clinical studies. And of course, they start here, but to develop the complete data, they need to have a very huge amount of clinical data looking at in immunogenicity. Whereas for a biosimilar, this pyramid is inverted. And the biosimilar development, the physicochemical properties is what is very, very important. And when you come to the clinical, this needs to be very, very small as compared to the innovator brand. So this is the key difference between a biosimilar and an innovator development. And that is why the costing for a biosimilar is much less. So we talk about the totality of evidence for a biosimilar, starting with the analytical studies, the animal studies, clinical PK/PD, clinical immunogenicity, and the clinical aspects. And at each step, they need to look at various aspects to see how similar they are to the innovator. So as clinicians, what should we look at to say a biosimilar is good for us to use for our patient? The common fears that are there are the fear of low quality and substandard product, similar but not identical, and there is where most of us have confusion, safety and immunogenicity, basis for considering efficacy, approved indication of reference product, not studied for many indications, and an important question of interchangeability. So how do you ensure the quality? The manufacturing process must comply with the quality requirements. And of course, the state-of-the-art process, knowledge, and understanding. And the two important things for extensive comparison is the physicochemical properties and the functional characteristics, because these are the two important things that ultimately matters to the clinical outcome when we use these for our patients. One more thing we must remember is that uh, when we talk about biosimilars, this is not a very new concept because the biotechnology has been there for some time, and important thing that we need to remember is each batch of the innovator is not identical. So truly speaking, the two batches of an innovator itself is a biosimilar. Small differences which can impact efficacy can be picked early in development, and that needs to be corrected. And how much difference we can allow depends on the heterogeneity of the product, and this should be justified and explained regards to the impact of the clinical outcome and the practice. And this is an example of the EMA approval of biosimilar erythropoietin alpha. Now, the safety concerns about biosimilars, we always talk about evidence and database. We always say that there is insufficient or very low data for these drugs, but that is not a concern, because what is important is the biochemical and the physicochemical properties that have been proved. Safety can be assured, therefore, based on the characterization similarity, the PK similarity, and the efficacy data. And the biosimilars have a low risk of new or CDS ADRs than the biologics that we have already studied. And the immunogenicity, of course, this is an important issue, and this needs to be an ongoing process, because this depends on various other factors which have crept in during the biosimilar that is being manufactured. It can be based on the cell lines that have used or some of the other ingredients that are used, but it should be in such a way that the clinical outcome is not changed in any way. So basis for considering efficacy is that we need to look at the study population. There should be homogeneous and sensitivity towards the effects that have been seen with the innovator, and the endpoint that needs to be based on unconfounded pharmacological aspects. For example, for a GCSF, you look at neutrophil count. For a FSH product, you look at oocytes. And the comparability margin that needs to look at. The largest difference in efficacy that won't matter in clinical practice. So that is what we need to consider, that when you use this drug in your next patient, the clinical outcome is going to be just the same as the innovator brand. The efficacy endpoints for anti-cancer monoclonal antibodies is overall survival. But this is not the ideal thing for a biosimilar, because then again, the cost of the drug is going to go up. So we use more sensitive endpoints that are considered today for biosimilars. Some of them are the objective response rates and the change in tumor mass. And the focus on detection of potential efficacy than efficacy per se is what is considered for a biosimilar, in contrast to an innovator when that is developed. Now, a word about indication extrapolation. Indication extrapolation, indications other than investigations done for clinical trials need scientific justification, but the following requirements need to be fulfilled. That is, convincing similarity with the reference product, and the clinical similarity in a key indication. So if a study is done in metastatic breast cancer, the similarity that is seen with the innovator in that indication is what is accepted. And unacceptable immunogenicity needs to be excluded for each route. So some of them may be an intravenous route and some subcutaneous route. So the immunogenicity for both routes need to be very similar. And the formal lack of a clinical trial in respective indications. So again, this is just summarizing how this is done. And a word about the nomenclature. Naming is important because for the pharmacovigilance of this, so what it contains is a non-proprietary name of biologic products. That is the initial part, following which a suffix can be anything with no meaning at all. Interchangeability is a thing that needs to be looked into, but we do believe that interchangeability should not be there because of certain changes in the process itself, which can produce increased adverse drug reaction or increased immunogenicity. So today, interchangeability is an issue with the biosimilars getting interchanged. So the clinical perspective of addressing concern, physicians are skeptical on the safety and impact on reimbursement concerns. Physician perception, which I've showed you, which is again a concern that needs to be made, and strong clinical data is important for acceptance. perception that switching to a biosimilar may not be good is again not the right thing to do. And what is important is to increase the use of these is the education that is essential for uptake. Unbiased experts focused on clinical data, national meetings and online education, and integrated into clinical practice guidelines as pathways. I think CMEs and conferences like this need to use these platforms to talk about this, because that is when the use of these biosimilars is going to increase. And as I mentioned, all the advantages of the biosimilars that are coming in will be reaching the patients, and the healthcare system will be taken care of. So there have been biosimilar guidelines across the globe in 2020, and this is the increase that's happened in 2016. In fact, India was one of the very early countries to have these guidelines on biosimilars, and we've been lucky, and we are all bearing fruit of several of these biosimilars that are there. And this shows you the number of biosimilars that are globally. You can see the graph for India is pretty high as compared to other countries. And you know the various reasons for this. This is just showing you the various drugs that are approved across. And to summarize, biosimilar should be highly similar to the reference product, as I mentioned, on the various characteristics, because that is what defines the clinical performance. The focus on biosimilar development is not established per se to convincingly demonstrate close similarity to originators, the processes that I've shown you, and these can be considered therapeutic alternatives to the reference product. With that, I will stop and thank you very much.